I started my career years and years ago, there was pretty much nothing other than animal research going on. But now we know that there's a very different stories emerge. We now have very much more sophisticated cell culture methods. We believe that human studies are essential. It's when we grow cells in human serum, we get different proteins expressed, we get different behavior of the cells, different structures of the cells and so on. So it's really important that we grow cells using human proteins, human serums and human cells, because that will reflect what's actually going on in the brain.
My grandma was actually diagnosed with cancer. So I was always, since little, very interested into the brain, how the body works, and I already knew that if I don't do medicine, I would be doing something close, so medical research. I was mostly interested in not involving any kind of animals, because that is what is most relevant to us.
8 years after I started working on brain tumours, my mother was diagnosed with a malignant brain tumour, and she died about 16 months later. So I suppose there was always that at the back of my mind, that I was doing this for a more personal reason. But of course, all the way along the line, I've had contact with patients, with carers. So it's very important, I think, that as scientists we engage not just with the other scientific community members, but also with patients, with carers, with clinicians and so on. And if anybody needed any excuse to carry on with brain tumour research, you just need to talk to somebody who's lost a child to a brain tumour, or has a brain tumour themselves, or have a loved one with a brain tumour. So it's not something that you have to fight for. It's something which is there almost, by nature of the beast.
It is very interesting because it's so new. Like the blood-brain barrier is quite in 3D. It's very interesting. It's challenging, but also it's like the drive to actually get something possibly unique out there. That is what keeps me going.
I think we've evolved a long way. We've even got a blood-brain barrier model where we can squirt fluids through and look at the flow of cancer cells and of drugs through the barrier model. But I think we've got these very sophisticated methods available to us, and that's gradually hitting the scientific community, and people are now being much more aware of these realistic alternatives to using animals. And they're also now becoming increasingly aware of some of the problems with using animals. So there are some profound differences between animals and brain. Over many years we've seen things being very effective in vitro, being effective in an animal model, and then failing dismally in a clinical trial. So it's not truly representative of the brain tumour it came from. The vast majority of drugs fail because they don't work in the human animal. So what we can do is we can start to remodel the human brain and the human brain tumour. We can then use that in 3 dimensions, and I think we'll be able to then more accurately decide whether the drugs that we're using will have potential in the patient itself. Thank you very much for supporting the work that my laboratory carries out. It's really essential that people like you support this work, because we wouldn't otherwise be able to do this. This gives us a unique opportunity to foster the work that we're doing, to pioneer new models, to develop new models, and to utilize these all-human approaches, which can be so critical in answering the questions about human diseases, which we can then hopefully treat in a far more realistic way.